morning to everybody. In the previous session, you already studied some part of the asexual reproduction. Now, today we are going to study about the vegetative reproduction. Plants reproduce asexually through their vegetative parts like root, stem and leaves. The new plants which are formed by vegetative reproduction, they are genetically identical to their parents. Vegetatively reproduce the plants by natural method or by artificial method. Artificial methods are used to propagate desired varieties according to human requirement. First, cutting. The small piece of any vegetative part of plant having one or more bud is used for propagation. In the cutting, stem cutting, that is, stem is selected for vegetative propagation. Example, rose, bougainvillea. Next, leaf cutting. Example, sansevieria. Root cutting. Example, blackberry. Next method, that is the grafting, in which seon and stalk, they are joined with each other. It is known as grafting. Seon, which is a part of stem containing more than one bud, and a stalk is the rooted plant, is called as a stalk, whereas budding is also called as bud grafting, in which only one bud is joined on the stalk. Example, apple, pear and rose. We will study this grafting in case of rose with the help of figure. This is the seon and this is the stalk. In the bud grafting, with the help of sharp knife, the bud they are cut and appropriate cut they are take on the stalk. Now this bud they are fit on the stalk, they wrapped or wind this bud on the stalk, cambium of the stalk and this bud, they joined with each other, and in the favorable condition, these bud they sprout to produce new plantlet. Now, next, tissue culture. In the tissue culture, small amount of plant tissue is carefully grown to give new plants. Generally, in the tissue culture, small plant parts they are isolated from the plant in the aseptically they grow on the nutrient medium and a number of plantlets they are produced. Micropropagation is one of the best method of tissue culture for propagation of plants. Now, sexual reproduction. The flower is a specialized reproductive structure of a plant in which sexual reproduction takes place. Typical flower which consists of four different whorls, that is calyx, corolla, androecium and gynecium. In the sexual reproduction, it involves fusion of two compatible gametes or the sex cells. In other words, sexual reproduction is the fusion of male and female gametes and the formation of zygote and embryogenesis. In the sexual reproduction, there is a genetically offsprings are produced which is not identical or they are dissimilar. There are sequential events that occur in the sexual reproduction are grouped into three distinct stages. First, pre-fertilization, fertilization and post-fertilization. In the sexual reproduction, these are two major events which is takes place. One is the meiosis and other is the fusion of gametes. The male reproductive whorl of flower is the androecium, and individual member of androecium is called stamen. Each stamen, which consists of filament, connective and anther. Now we will study about the structure of anther. Each anther which is represented by a group of parenchymatous tissue surrounded by single layered epidermis. And anther, it may be monothecous or it may be dithecous. In case of monothecous anther, there is a presence of only one anther lobe, only one anther lobe, but having the two pollen sac. In case of the dithecous anther, which is having two lobes, but they are having four pollen sacs, and therefore it is also known as tetrasporangiate. Now we study about the tiers of anther. During differentiation of some hypodermal cell get transformed into archisporial cell. Now these archisporial cell, they divide into inner cell called sporogenous cell and outer cell called parietal cell. Now this inner sporogenous cell, they form the sporogenous tissue, It is also called as microspore mother cell, which is diploid. It undergoes meiosis to form microspore tetrad. Now this parietal cell, they forms anther wall layers. There are four anther wall layers. First epidermis, second endothecium, third middle layer, and fourth tapetum. Now all these four layers we will study with the help of diagram. Now in this figure, epidermis is the outermost layer. Single layered cells are barrel shaped and which is protective in function. The second layer, that is the endothecium. Endothecium, they are present below the epidermis, which is also single layered, but the cells, they are somewhat larger. And in this endothecium, there is a presence of fibrous thickening. These endothecium, they are helpful for the dehiscence of the pollen sacs. The third is the middle layer. The cells are thin, 2 to 3 layers, and at the maturity, the middle layer, they are disintegrates. Innermost to the middle layer, that is the fourth layer, is the tapetum. Tapetum, which is also single layered, and this tapetum, which is a nutritive tissue. Below the tapetum, these are the sporogenous tissue, centrally placed sporogenous tissue, in which it is also known as microspore mother cell, which is a diploid. When the anthers, they are mature, at that time the sporogenous cell, which undergoes meiosis, and each spore MMC, they produces tetrad of microspore. Now this is a dihyst anther. At maturity, the pollen sacs, they are breaks and pollen grains, they are released. Thank you.